is Jinghong Cui. I'm a freshman from Duke University, and I'm an ambassador of DKU Bio110 class, taught by Zhiyong Li, a professor from Duke University. And I'm going to introduce our class and also one of our topic, mitosis. And first, I'm going to talk about our class, DKU Bio110. It's an integrated science biology that employs 5 themes that describe properties of life and will be reiterated over again in integrated science biology. The first one is organization, which include structure and function. The second one is the cycling of energy and matter, and then it's information, mainly is genetic variation, and then it's homeostasis and evolution. And the themes will be unified under the organizational principles of the scientific methods, formulating hypotheses and tested with experiment. Students in integrated science biology are develop the understanding of key concepts in the context of cross-talking with chemistry and physics. And then my presentation is divided into 4 parts. The first one is introduction to mitosis, and second one is stages of mitosis, and then mitosis in our lab, and the last one is mitosis in medical and research context. And first I want to introduce what is mitosis. And mitosis is a type of cell division that results in 2 daughter cells, each having the same number of chromosomes as the parent nucleus. This process is crucial for growth, development, and repair in multicellular organisms. And then I'm going to introduce the stages of mitosis. And first one is the interphase, and it is about DNA replication and preparation. During the S phase, DNA replication occurs, ensuring each chromosome has 2 identical sister chromatids. And then is the G1, G2 phase involves cell growth and preparation for division. And actually, is first one is G1, and second is S, and then it's G2 phases. And then is the prophase. During this period, chromatin condenses and becomes visible as chromosomes. Each chromosome contains 2 sister chromatids, and the centrosomes move toward the 2 poles of the cell, and the spindle begins to form. And the nuclear membrane gradually disappears, and microtubules of the spindle attach to the kinetochores of the chromosomes, and the chromosomes move further towards the center of the cell. And then is the metaphase. All chromosomes align on the equatorial plane of the cell, forming the metaphase plate, and the cell is in the most stable state of division during this stage. And then is anaphase. Sister chromatids separate and move towards the opposite poles of the cell. This process is achieved through the contraction of microtubules. And finally is the telophase and cytokinesis. In the telophase, the chromatids that have reached the 2 poles of the cell gradually condense, and a new nuclear membrane forms. The cell changes its shape in preparation for cytokinesis. And it is different in animal cells and plants. In animal cells, cleavage occurs through a contractile ring, while in the plant, a cell plate is formed. Eventually, two independent daughter cells are produced. And then I'm going to talk about medicines, our lab. One of our lab is about the observation of garlic root tip cell metastasis by optical microscope. And the experimental step, first of the experiments, we cut about 1 centimeter long roots from the vigorously growing garlic root tips and soak them in the fixative solution for 3 minutes. Then use forceps to select 4 to 5 roots and place them in a watch glass. And then add 4 to 5 drops of 1 mol each cell and let the roots tips soak in the acid for 5 minutes. And then we gently rinse them 2 to 3 minutes, 2 to 3 times with clean water, and then add 2 to 3 drops of 0.8% gentian violet staining solution to cover the root tips and stain for 3 minutes. And then we rinse out the excuse staining solution with clean water. Place one of the three root tips on a clean slide and add a drop of distilled water and press the cover slip with the eraser to make it into a single layer of cells. And finally, we use a low power objective of the microscope to find the cells, and then switch to the higher one to observe the cell structure and the chromosome in each stage of mitosis. And here's the picture of our observation. The cells of different period observed under a microscope in the laboratory. And in the interface, the cell nuclear appears round or oval, and most cell in figure 1, this one, is in this period, and the yellow circle one is the typical one of this stage. And about the process, the chromosome gradually condensed into structures, and the yellow circle in this picture, figure 2, and the figure 3, this one, is about this phase. And in the metaphase, chromosomes are highly condensed and neatly arranged on the equatorial plane in the center of the cell. And we can see it clearly in the fourth picture here, in the red circle. And in the anaphase, the centrosome is divided, and we can see in the picture 3, the red circle, it is clearly divided into 2 parts. And finally, it's the telophase. The chromosomes reach 2 poles and start to be decondensed, gradually returning to the state of the interface. And we can see clearly in the red circle in the figure 2 and the blue one in the figure 3. And finally, I'm going to talk about the mitosis in medical and research context. First one is the cancer diagnosis. It's about mitotic index, and the mitotic index, or we can say it is the ratio of cells undergoing mitosis, is a key indicator in cancer diagnosis when this index is high and is often related to the aggressive tumor growth. And also the drug of cancer. Many cancer drugs target proteins involved in mitosis, and those drugs aim to disrupt cell division and inhibit tumor progression. And then is the stem cell research. It's a role in regenerative medicine, and mitosis in stem cells is crucial for tissue repair and regeneration. So understanding mitotic control in stem cells can lead to new therapies for degenerative diseases. So as we can see, the mitosis is very important in our life. And here's the references in this PowerPoint and my presentation. And that's all for my presentation. Thanks for listening, and welcome all DKUers to come to take June Lee's Bio110. and also welcome everyone to come to DKU to have some academic exchange on biology. Thanks.